Next up, I believe we have cucumber mosaic virus with Dr. Escalante.
Good morning or good afternoon, everyone. I'm Cesar Escalante. I'm a clinical assistant professor and a vegetable extension pathologist at Purdue University. And today we're going to be talking about CMV, a very important virus, and in the cucurbit industry. And today, this is a very prolific virus. It's a very economically damaging pathogen in vegetable production, and not only affects cucurbits, but it also affects other vegetables. But obviously, we are talking about cucurbits here. So, cucumber mosaic virus or CMV was actually first identified in the United States around 1916. This is way before the era where plant virology was well developed and where people were using molecular or even electron microscopy was not well developed at that time. But there were some reports in Michigan and also in New York, what they call mosaic disease of cucumber. They later found out that this was actually CMV. So CMV has been around for a long time. And so this basically is a top tier threat because it has a massive host range. It has over 1,200 species that can host this virus, and it affects many crops. But like I just mentioned, but it's important in cucurbits, especially in cucumbers, melons, and also squash. And those three type of crops is very devastating. And so we're going to talk about symptoms and other type of characteristics of this virus, especially how this spreads and things like that. So CMV, it's everywhere. It's not only in the Americas, it's pretty much everywhere in the world. And in the United States has a nationwide distribution. It can thrive in temperate, subtropical, or tropical climates. And this map, I just highlighted some of the areas where a lot of cucurbit production happens in the United States and where we get most reports of CMV being present. That doesn't mean that it's just located in those states. It can be pretty much anywhere where cucurbit crops are planted. Well, it's more famous for attacking cucumbers and muskmelons and frequently hit summer and winter squash, pumpkins, and gourds. It can also affect watermelons, but with less frequency. There are just a few strains that might affect watermelons. So the virus success is due to a strategy that they use of using reservoir weeds. Basically, it overwintering in perennial weeds, like for example milkweed or chickweed. They wait for the spring aphid migrations and then begin the cycle again. That's the strategy that basically the virus uses to be able to survive. And what does CMV look like in the field? Well, you basically have to look for the mosaic, just like the name says. So the classic mottle part and the light dark green of the leaves that you will also see some a pinasty they call it. As you can see this here, basically it's a downward symptom on leaves that become distorted leaves, basically. So that's the most common type of symptoms. And then what we can see here is that typical white pickle, they call it for the fruit, and especially in cucumbers, that it becomes pale, smooth, and distorted, that then develops sometimes these dark green warty bumps. That's a very typical on cucumber fruit. So what another important thing I want to mention about symptoms is that if infection happens early in the season when plants are very small, the plant will be severely stunted, and often that looks like a bushy rosette, like in this picture here in the panel B. And in some cases, in melons, for example, if the fruit develops or the plant is infected, the fruit might develop this bitter taste, making the crop basically unmarketable, even if the fruit look okay. So that's an interesting feature of this virus. So symptoms typically appear between 4 to 14 days after infection. That will depend on the environmental conditions and also the host, basically. So in terms of diagnosis, we have to basically start with visual scottings. That's the great start here. But just like Carrie mentioned, CMV symptoms can look like very similar to other viruses. This is very common with all plant viruses. Sometimes CMV symptoms can look like zucchini yellow mosaic virus, for example, or even herbicide drift. And so if you want to have a definite answer, we need to rely on two main tools. In this case, it can be a serological test. We have two options, it's ELISA or immunostrips. The advantage of immunostrips is that you can get results in about 30 minutes. You have the versatility to take this test to the field also, so you can get a quick result. And basically, this test will detect the virus gut protein. That's what it does. It's very cost effective and very common. But if you want to have higher sensitivity, we use RT-PCR. This is an RNA virus. Of course, we have to do RT-PCR and to be able to detect the RNA itself. So that would be a more reliable kind of test. And I will say that most of the diagnostic labs in the United States offer these services, so I will highly recommend using them before making major management decisions. So I will say that the primary engine of CMV spread is the aphid. There are more than 80 species of aphids that can carry the virus, and the transmission it's in a what is called a non-persistent manner, which is a fancy way to say it is a lightning fast process or way to transmit viruses. And basically, an aphid doesn't need to colonize a plant. It only needs to probe and or taste the leaf for a few seconds to pick up the virus in its mouth part or style it. And then it will move to other plants and will start infecting or transmitting the virus immediately. So because the virus stays in the stylet, it doesn't enter the aphid's body, so the insect only remains infectious for a few hours, but that's more than enough time to infect the entire plant, or even several rows within a field. Another important feature of this virus is that it's not only transmitted through insects, but also can be transmitted through mechanical manners. And in cucurbits, it's very common that we do several harvests during the season. And that's a good way to spread the virus, especially when you're using tools that will cut the fruit and then you move on to the next plant. So if you have an infected plant and then move into a healthy plant, that's a good way to transmit the virus. The good thing about it, from my observations, is that once the plant it's at that later stage, the effect of the virus will be very minimal if it gets infected. But sometimes in some crops there are some cultural practices like vine turning, for example, and that's done usually early in the season or mid-season. And in those cases, if there are plants that are infected, and then you move on to healthy plants, that most likely you're gonna get infection, and that at that stage the problem can be more severe. So there are several epidemiological factors that favor transmission or infection of CMV. And sometimes we ask, why some years we see massive outbreaks in viruses, and there are several reasons. It is basically a combination of three things or three factors, like for example, weather. It can be vectors or weeds, because weeds serve as the host for many viruses. So sometimes mild winters, for example, will allow more weeds to host to survive. So that provides a larger virus bank for the insects. And sometimes we have warm springs that accelerates the aphid reproduction. Then we might have more disease presence in the field. So CMV often occurs also in mixed infections. Carrie just also mentioned that it's very common with viruses. And when a plant has CMV plus another virus, like for example, a common combination or mixed infection that is found is CMV and watermelon mosaic virus. The symptoms are not just double, but sometimes can be just basically magnified. And that synergy leads to much faster plant decline and definitely a total yield collapse. And then when we talk about disease management, I always say, well, how do we fight back? And I always say that the the first layer that you should have is resistance. many of us know that there is no cure for a virus. Once that plant is infected, no management, you can do it will basically prevent from the spread. So the management is 100% prevention. So genetic resistance is the number one defense. Always look for that label in your seed that has some sort of resistance, even if it is intermediate resistant or complete resistant. But it is good to make sure you are planting seed that has some sort of resistance. In that way, you have that first layer there. And then you can also implement other practices like cultural controls. For example, using reflective mulches that basically disorient the aphids from and prevent them from landing on your fields. Um, Also practice sanitation. If you see an infected plant, pull it up and bag it immediately and try to take it out of the field. If you don't touch plants that are healthy if you touch an infected plant, because like I say, this virus is transmitted also mechanically, so you will get your all the healthy plants infected if you do that. So another Another important thing to have in mind is that sometimes if we see aphids, we tend to apply insecticides. And it's very common that when viruses are transmitted in a non-persistent manner, spraying for aphids often increases the infection of the virus because the spray irritates the aphid. That causes them to basically move and prop more plants because the aphids are just distorted. And then that basically leads to having more plants infected. So I would say focus on weed control because those are good hosts for the virus and resistant varieties instead. And always talk to your local entomologist and see, what are the best options in terms of managing aphids in these particular cases. I just want to mention that if you want to visit our website here, there is a list of all the members, and if you have any particular questions about this virus or any other viruses, just feel free to reach out to us, and we'll be happy to help. And this is just a lot of support for my program, and I'm very thankful for the emerging viruses and Cucurbits working group for letting me be part of this webinar series. And thank you for your attention.
All right. Thank you, Cesar.